So welcome to this free webinar on sequence to structure. This is part of an ongoing OmicsLogic Genomic program, which we had started on March 12th, 2021. We will focus on how to connect the various elements of analysis of genomic data to insights that are actionable in context of a particular project. My name is Bipsha Biswas, and I'm the OmicsLogic Community Manager working closely with participants who join us from around the globe, from different academic and research backgrounds. This program is part of an OmicsLogic Bioinformatics training program for infectious diseases that started in January 2021 and will continue until July 2021. The first part of this program was on infectious disease and focused on the use of bioinformatics and big data for infectious disease research, diagnostics, as well as drug and vaccine development. This webinar is part of this second session, which is OmicsLogic Genomics. In this second phase of the training program, participants are learning about concepts and analytical tools used to study genomic variation in infectious diseases. Each week, participants are meeting online to discuss various aspects of NGS data processing, genomic data analysis, and interpretation. In the next phase of this training program, we will focus on RNA-seq analysis. This training will include methods of machine learning for bulk and single-cell gene expression studies, special focus on examples for infectious disease research, such as malaria, COVID-19 disease prognosis, immunology, and biomarkers. So this program starts on April 16th. Now that I've given you a background of this webinar that we are having today, let me talk a little more about what OmicsLogic program is for those who have joined us for the first time. OmicsLogic is an international program which is running in 5 different regions with over 10,000 users across the globe. Due to this fast growth, our company is working with the local and regional coordinators who are helping refine local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. The OmicsLogic programs follow a project-based learning approach, which we are going to be talking more about today in later part of this webinar, using research-grade tools to analyze data from top peer-reviewed journals. This OmicsLogic training has been completed by over 10,000 participants from 162 countries in over 300 workshops for 6 different specialization tracks, which include oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. In the upcoming program, you will have the opportunity to interact and collaborate with an international team of experts specialized in the application of data science to biological data sets. Let me introduce you to the program mentors, who many of you may have already interacted with. We have Dr. Harpreet Kaur as our certified OmicsLogic trainer and expert bioinformatician, who guides these hands-on sessions and assists participants with projects and analysis during each of these OmicsLogic training programs. Her area of specialization is in genomics and machine learning. Dr. Mohit Mazumdar is our OmicsLogic project mentor and an expert in big data computational analysis. He has a PhD in computational biology with over 10 years of experience working with industry and academia. Ilya Brodsky is the CEO and co-founder of Pine Biotech. In his current role, he has extensive experience working together with industry and academic partners who are applying multi-omics integration bioinformatics to a variety of biotechnology and clinical applications. I will now like to invite Ilya to talk more about sequence to structure, which is a topic for today's webinar. Over to you, Ilya. Thank you.
Thank you very much, Bipsa, and welcome everybody to this webinar. So before we start, let me introduce myself. As Bipsa mentioned, my name is Ilya Brodsky. I'm the co-founder and CEO of Pine. And our vision here at Pine Biotech is to enhance human health and well-being by enabling biological research and discovery with relevant data, solutions, and expert support. And so one of the questions that I want to talk about today is how can bioinformatics actually assist in this vision? So to clarify, the mission that we have is to simplify bioinformatics and advance various research projects through our modular and intuitive multi-omics analysis platform that is powered by both human experience and artificial intelligence. And what we do is we offer training, research support, and big data analytical tools that are designed for research and biomedical discovery. So today, I want to talk to you about how we will be able to leverage genomic data to arrive at meaningful and actionable results for infectious disease. And so this is important because I want to focus on this aspect of actionable. So how can we make research in infectious diseases using genomics into translational research that could be applied? So the basic question is, given the availability of genomic data, the widespread data collection and analysis efforts around the world, what specific issues are hindering or preventing the translation of this research into action? And so to talk about it, I want to focus on the specific elements of bioinformatics that can help us align data using a variety of references that we have to be purposeful about selecting, how we question the data and what specific aspects of the relationships we focus on, and then how we can translate that insight on the sequence level to the structure, and how we can model and analyze the interaction between different proteins to improve our models. And so here are some specific things that we can discuss in the context of basic to translational research. So in the beginning, we typically focus on hypothesis generating research questions. And so a lot of these questions are really to find patterns, explore their consistency and significance, and start focusing in on a particular element of that variation. Then we go to hypothesis testing, where this hypothesis that we've generated from maybe a small collection of data, we want to test whether it is translationable for the whole population. And here we have to define what is that population that we are interested in. And then finally, we want to think about what are the actionable insights that we can extract from this knowledge. And so if you kind of think about it, from getting an idea to specifically generating knowledge that kind of helps put that idea into the proper context, that is essentially what is needed for action or for the implementation of that knowledge that probably has a lot to do with communicating and convincing other people of how this particular insight is relevant to an intervention, maybe increased data collection, or maybe a long-term strategy that can involve multiple people that have to work together. And so the basic question again remains, how can we translate this information from bioinformatics analysis of genomic data to a translational impact? And so if you look on the American Society of Molecular Data, you can find the definition for translational bioinformatics, which is the development of storage, analytic, and interpretive methods to optimize the transformation of increasingly voluminous biomedical data and genomic data into proactive, predictive, preventive, and participatory health. And so what defines bioinformatics in the era that we are in is that we are seeing an increasingly huge volume of complex data that has a lot of variation, as we will see in today's session, into clinically useful knowledge that requires the convergence of molecular bioinformatics, biostatistics, statistical genetics, and clinical informatics that are translated into computational, easy-to-use methods that can be used to establish a better or a more practical and accelerated path from discovery to improved care. And so along the way, it's important that the discoveries become practically useful. So to achieve this goal, we are essentially looking at the advancement of translational genomics that are going to turn this large data or big data into some practical questions that could be answered and can have a major impact. And so before we kind of go on, I want to highlight some of these elements of what translation might look like. And so one of those is identifying flawed science. So a lot of times scientific publications include a lot of different data sets, and it's often easy for us to see it from outside, but it's difficult when you're a part of a research project to understand the biases that drive the research question. And so these biases could be many. And I want to ask you here, I'm imagining that many of you are involved in some kind of research. So what are some of the biases that you might imagine that drive flawed science? So what are maybe some of the reasons why we have flawed science? What do you think? Okay, so to answer the question, you can write your answers in the chat. So hopefully we can have a discussion about each one of these points just really briefly. So what do you think might be some of the things that could drive flawed science? Okay, so Aina is saying poor statistical sampling numbers. Okay, so incorrect data, right? Selecting bad data, poor data. Okay, great. Thank you, Sanjeev. What other things? Okay, gap between understanding the data and interpretation. Excellent. Technical errors, batch effects, that includes technical errors, batch effects. Okay, high error rates, incorrect data, maybe data that was incorrectly collected. Excellent. So good question. So essentially, okay, so now we'll get to methods here in a section. So wrong assumptions. Okay, excellent, Ayo, thank you. So we can identify flawed science by revisiting the research projects that have been published, looking for gaps and limitations that led to incorrect interpretation, right? And so that is a big part of translating our efforts into something practical. Identifying that flawed science essentially leads to new questions about how that particular assumption could be addressed, adjusted, improved, and ultimately lead to novel insights. Now, the other part of it is how can we improve our estimates? And so somebody already mentioned here that we have proper methods to analyze data, right? So we operate by having estimates. So we estimate the rate of mutations. We estimate the level of expression. We estimate the frequency of occurrence. Those estimates are based on our understanding of the problem. And so to improve those estimates, when we come at a problem, we need to collect more data that will lead to better estimates, right? So again, if we kind of think about it together, what do you think are some examples where better data or better contextualized data can improve our estimates? Okay, so if anyone wants to, again, pitch in, just put your answers in the chat box. So how can better data or better contextualized data improve estimates? Okay, so Swathi is answering data cleansing and class labeling can be done. Minimize deviation. Okay, so let's imagine, for example, the current situation with the pandemic. With the pandemic, we have millions of samples being collected from around the world from the COVID-19 pathogen, right? And so the question is that sampling consistent throughout all of the regions? Can we improve our estimates of emergence of novel mutations and variants of concern based on better data that is collected in a more precise way from around the world, right? So for example, if I have, oversampling in the US and undersampling in Argentina, could we improve the balance and so get a better picture of how this virus continues to evolve, right? So those are some of the questions that we could, again, improve by looking at the quality of sampling. Thank you, Leanna. Taking advantage of larger data, right? So sometimes we only focus on a small portion of data, and that leads us to ignore the information that we have from the rest of the data that we have available. Sometimes it's the opposite way. We want to take a look at everything, but we are ignoring the quality of the data. And so instead of looking at the overall trends, we're biased by this incorrect data. Okay, so Sanjeev is mentioning using the same platform for analysis. Maybe this means that we're only looking at one particular type of methodology, or maybe we're looking at one particular type of sequencing, for example, or data collection methods. All right, so thank you for your insights. That's really good to hear. So now let's talk about how we can infer new insights about mechanism of disease. So again, we have typically this textbook knowledge of actual disease mechanisms and patterns of progression. And so a lot of times that is because the knowledge accumulated over years gets internalized and translated into these static models, right? And so we say that typically a virus enters the cells, starts replicating. Inside that replication, there's a certain machinery that is hijacked, and eventually we have this progeny of a novel virus. But in reality, this is a highly dynamic process. And so instead of basing our understanding on just a static model, we are moving towards dynamic modeling of various aspects of mechanisms of disease and disease progression that have to be generated through data. And so a lot of that goes back to the data that is collected, that is being analyzed, that is being converted into a dynamic model that could be refined from patient to patient, from country to country, from infection to infection. And so some of these elements here on the screen, I think, should give us an idea of how challenging translational bioinformatics really is and what kinds of efforts are needed to make specific research questions answered in an actionable way. And so that is one of the reasons why we are thinking of how we can develop a roadmap for translational bioinformatics that includes a process of helping researchers, scientists, and students find relevant data sets that are available for public use, how to properly explore and visualize to get a sense of what kinds of variation are standing out and are critical and interesting for further research. We prioritize a development of research methods that are easy to use and compare, and also explain the basis of these research methods so that they are used with an understanding. And ultimately, we are looking to connect the data with the insights and understanding through this process of translational research. And so one of the programs that OmicsLogic developed for infectious diseases is this program called Bi-Informatics for infectious diseases. And so just to give you an idea of what kinds of topics were covered so far in this program already, this program is really intended to help guide the understanding of pathogens and disease that they cause by looking at zoonotic transmission, viral evolution and replication, as well as bacterial and parasite infections. We also talked a lot about how to collect genomic, transcriptomic, and metagenomic data that help us understand and contextualize disease, and apply that to the questions arising about the emergence and the intervention of these disease, including vaccine design, antiviral drug design, and antibiotic resistance, as well as epidemiological trends and drug resistance. And so we also do this using projects where we take a look at recent epidemics like Ebola, Zika, and dengue. We look at coronaviruses that are being talked about so much now, Mycobacterium tuberculosis and leprosy, as well as malaria. And so as a result of this program, we are really trying to address this important aspect of infectious diseases that have a prevalence around the world and are causing about 11 million deaths per year, where the impact primarily is focused on mortality, but very specific interventions that are driven by people that are in the position of some of you here that are present and us that are interacting with researchers at various academic centers can really make an impact. And one of the primary ways that we could address this growing challenge of infectious diseases is really change the way we think about the impact to only be preventative or intervention in terms of treating that disease, right? Because there's such a disconnect between the research and the actual practical action that could be taken as a result of those insights. And one of those examples is just simply surveillance. And I think that we can see how that is being done today. So in the context of COVID-19, we saw that no surveillance, as opposed to surveillance with a purpose to detect, identify, and explore data sets that are being collected in an active way, can lead to a significant reduction in the number of cases, in the number of deaths from those infections. And so you can see here the number of cases that increases as a result of no surveillance and decreases with passive, and even decreases even much more with active surveillance that is linked to inaction. And so that is one of the things that we want to talk about today in the context of some of the projects that have been discussed in the context of this Bioinformatics for Infectious Disease training program. So in the program, we decided to focus our attention on infectious diseases, and those are primarily categorized by diseases that are caused by pathogenic microorganisms. So because we're focused on infectious diseases caused by these microorganisms, we started talking about specific examples that are very relevant to these diseases around the countries where we are working on this training program. So for example, bacterial diseases, viruses, and parasites all cause very different types of manifestation of disease. And even though they all trigger the immune response, we have to contextualize our understanding and the data. And so we've had to learn a lot about the life cycle of the malaria parasite and how the specific stages of the disease progression in malaria, for example, plays a role on a variety of interventions. And so having come from a bioinformatics perspective to this problem, we can really see how there's so much data available on various pathogens that cause human disease that we can essentially learn through data to understand the interaction between the host, pathogen, and the vector to understand how the bioinformatic analysis could lead to impactful insights. And so after we've kind of reviewed the background of these different diseases, we've come up with a number of research examples that can demonstrate how we can apply data analysis to address the specific topic. So here I want to talk about a few examples and kind of demonstrate some of those in a few minutes using some practical tools. I don't know, Mohit, if you're here, if you could paste the link to the OmicsLogic website, and we can see some of the examples of what these projects really look like. So on the OmicsLogic platform, and I think this is something that we are not, okay, thank you, Mohit. We're not going to be able to go through completely in today's session, but it's important to understand how these specific diseases are manifested as a result of particular pathogens. And then what kinds of questions could be asked about these pathogens so that we can apply bioinformatics analysis methods in our analysis to gain some insights, right? So for example, what we covered so far was how we can categorize protein function and how genomic data variation could be a way to look into various immune pressures that are affecting the genomic variation of COVID-19. And so describing these issues, we can really start to focus in on particular topics of interest. For example, on how we can study the replication and colonization of pathogens, how we can study the impact of that colonization that leads to cell death or organ function and interference. And so to talk specifically about some of these examples, I want to first of all talk about what is this genomic data and how does it help us understand these pathogens. So first of all, the importance of genomic data in infectious diseases cannot be underestimated. We are really looking at an unprecedented look at the diversity and the variability of pathogens throughout their development cycle and the cause of disease at a single person, individual, as well as the whole community or a country, a region, or the whole world, right? So we are looking at unprecedented amount of detail. And so when we think about that detail, we can look at the viral evolution, how the pathogen spreads and causes disease as it adapts to a population. We can look at the interaction between that pathogen and other maybe less harmful pathogens that are competing for resources and domination, right? And we can also look at the host response. And so all of these areas of research have been profoundly affected by the emergence, availability, and the lower costs of genomic data. So here's an overview of how in the last 20 years, this availability of genomic data has led to an increasing understanding of how cells function at the molecular level and allowed us to understand the major causes of some of these diseases in terms of their specific mechanism of interaction. And so one of the things that we have to do to really utilize this data to gain those insights is we have to turn to the methods of analysis. Because ultimately just raw data cannot be used in any way to actually make any kind of conclusions about the pathogen itself. So right now I want to give you an example of a particular project that we will just briefly take a look at using the Tbio platform. So if you could go with me to server.tbio.info, and I want to demonstrate one of the pipelines that will combine several different analysis methods so that we can start looking at the output from this pipeline and think, how do we interpret the results of this analysis? So thank you, Mohit, for posting the link. Bipsa, if we have the demo accounts here as well, for those of you that do not have them, please let me know if you're on this link already by putting in number 1 in the chat box, so I'll know that you're here on the platform. Okay, thank you, everyone. And if Bipsa, you could share the demo details for those that don't have access.
Yeah, Bipsa is in another meeting, so let me find it and share it in a while.
Okay, thank you. So what we'll do here is we will take a look at one of the demo pipelines that helps us understand the translation of genomic data into a particular insight. So please go with me right here. Yes, Parissa, we are, Mohit is just getting those details for you right now, but I will just start the demonstration and then Mohit will share the details in a second. So I will go right here to this section under virology. Again, if you think about contextualization, the methods as well as data have to be contextualized so that they are easy to use to ask specific questions. And so this whole section about virology has been developed to assist with that process. All right, thank you, Mohit. He has shared those details. So I'll just give it another couple of minutes. If you have logged in with the demo account, or if you have your personal account information, please log in with your personal account. If you don't have an account, please log in with the demo account, and then I will continue to speak about this section and show you a couple of examples. So if you have logged in, put number 2 in the chat box. All right, thank you, Leanna. Marie. Okay, let's give it a minute until everybody else is in. Okay, thank you, Parissa. All right, great. Excellent, thank you, everyone. So we'll go right here to this section, virus quasi-species, and I will open this drop-down right here. So you can see in this drop-down that there are several different demos. And essentially, these are data sets that have been already analyzed, and we'll just take a look at what are the methods to arrive at that analysis result, and then we'll see how to visualize that analysis result to interpret it and think about what might be the value of that research question. So we'll look at this first demo, virus MSA and phylogeny. And when you click on that, you see this interface that essentially combines together several different methods of analysis that could be relevant to various types of input data from genomic sequencing. So one of the issues with genomic sequencing is quality control. And quality control is for next generation sequencing, essentially comes down to the quality of the actual sequence reads, including different technical adapters that should be eliminated. And so you can see those data pre-processing steps right here. Then we have some options for mapping. So mapping onto the reference genome if we have raw sequencing reads. With those raw sequencing reads, you can do a number of downstream analyses, including variant calling, the mutability or how frequently we observe mutations at the consensus and the primarily sub-consensus level, differential variant calling when we want to compare groups of samples, and then we also want to sometimes estimate time course dynamics of mutations. So then we have a specific section here related to not point mutations, but whole deletions of viral genomes. This is specifically for viruses. And then here we have a number of additional multiple sequence alignment and haplotype detection for inference of phylogenetic relationships, as well as the coding mutations that could impact protein-protein interactions. And so all of that is right here. The way that this section is organized is you can click on Start, and we will take one example to just kind of think about the implications of a particular question. So this example is taken from a paper about the emergence of SARS-CoV-2. And as you know, up until today, this question remains: what is the actual origin of SARS-CoV-2? And how can we infer the dynamics of the spread of a pathogen like SARS-CoV-2 to go back and trace it to a potential original host? And so we'll take this data set, and you can click on OK to go to the next step, and then you will have a number of one actual step highlighted as the next possible step. So now we'll do multiple sequence alignment. So we've loaded some data. This data has to do with SARS-CoV-2. We want to perform multiple sequence alignment and essentially find the specific variants that could be interesting for the next question that we want to ask. Now, at the genomic level, these mutations oftentimes do not translate to a change in the amino acids, so they're so they are silent mutations. We don't observe any change, and then we want to identify how some of those mutations actually do have an impact on amino acid sequences. And so here we have a specific example of different amino acids that have different chemical properties. And so now we can click on OK. Select codon position, because viral mutations only occur more frequently in specific codons. For example, here you can see some of the examples of which positions cause a change in different codons. And then we want to use all of the phylogenetic methods to infer the relationships between sequences by reconstructing a phylogenetic tree, or thinking about how did these viruses emerge from some common ancestors. And so how is the observed variation in the data that we have a representation of some kind of a evolutionary relationship? And so you can see an example here of how we can reconstruct this phylogenetic tree. Now, since we're going to be using multiple methods of relationship using phylogenetic tree reconstruction, we want to then analyze these different trees to think about how we can think of the real kind of molecular dynamics that happen in the evolution, as opposed to just looking at results of a particular method, right? So if we are just following one methodology, we are oftentimes biased by that methodology being developed to answer specific questions. And here we want to analyze all of the various results to identify a common trend. And that common trend is more likely to be representative of the actual biology that we're going to be looking at. So there's a method called tree space. Essentially, we'll analyze the different trees and help us decide what are some of the most representative trees of what's actually going on. So I'll click on OK right here. Now, to finish this pipeline, I'm going to click on End, and I will see this result for download pipeline output files. And so we're not going to necessarily look at all of these files, but we're going to take a look at one of the example files that will help us think about the translation of the insights from this analysis from a very specific data set that we have collected to ask downstream question. What is the impact of the identified mutations, and how can we use our understanding of the relationship between different viral sequences to ask the question, where could they have originated? So here you have different types of outputs like phylogenetic analysis. So here you can see the multiple sequence in FASTA and Clustal. You can see a visual representation of the tree, the phylogenetic tree, using multiple different methods of reconstructing that phylogenetic tree. And you also will see how that tree will be done separately for group 1 and group 2, because the input files that we had are organized into groups. But one of the important files that is right here is called full table Tsv. So let's take a look at this table. So you can download, it's a fairly large file, but you can click on it and we'll start downloading. And we'll take a look at what all the information from this analysis could be available for now asking specific questions, right? So I'll take a look at this and I'll open it in Excel just so that we can visually see what's inside this data. And please let me know, all of those that are following along and doing these steps, if you can actually download and open this file on your computer to just kind of see what's in there. Okay, maybe you can put number 3 in the chat box if you were able to make this pipeline and get the same results. All right, thank you, Anonymous. Has anyone else gotten this file? Okay, I'll just give it one moment. All right, thank you, Nigel. So again, you can see some of the steps that we took. If you just scroll down right here, we went from loading some genomic data, performing multiple sequence alignment and mapping nucleotides to amino acid codons. And then we performed a number of phylogenetic analyses to create a representative phylogenetic tree. And so now we want to look at this one file that kind of combines this information together to start asking the questions of how do we analyze what's in the analysis results to get some actionable insights. So how many of you have this file on your computer and can see what's inside? All right, thank you, Marie. So again, if you are just joining, we are repeating this pipeline just to kind of see what are some inputs and outputs and think about what is the translational value of such a data set. Okay, thank you, Sarahni. Okay, let me explain what is in this particular data. Here you have the position on the reference genome that is followed by 5 different columns that indicate, first of all, this is the sequence of nucleotides. Here, the sequence on the positive and the negative strand of amino acids. Then you have some alternatives for the positive and the negative strand. Now, typically this kind of an output would be useful for DNA reference genomes, right? Then you would have to do, then you would have to go through both positive and negative strands. But in our case, it's just an RNA virus. And here you can see an example of how this particular sequence is different from this sequence in the reference genome. So here we have, for example, a mutation, right? There's a T instead of an A, right? If the reference genome contains a certain sequence, we can see here the genomic variation that is observed in different sequences. Here we see a lot of deletions, right? So as we look through this data, we can now scroll down a little bit and we'll get to a translated portion. And this translated portion includes the translation of codons. So these are trinucleotide codons into a particular amino acid, right? So now after we go through this, we can actually see that the picture is becoming more and more complex. We have the reference in nucleotide and amino acid. We have multiple sequences. And for all of these sequences, we have thousands of positions. So over 30,000 positions, right? So the question is, with the availability of all of this data, what are some of the questions that are coming to mind in the context of this project? So if anyone has any specific ideas, what might be the questions that we can ask about this particular analysis result? Okay, just by seeing what's on the screen or seeing this on your computer, you see all of this data, right? We have 30,000 positions through this reference genome. We have a bunch of samples that we can look up. For example, we have these different IDs that we can probably look up on NCBI. How do we interpret this kind of an analysis? What is the value of this analysis? And how do we quickly get from where we were with the raw input files to some kind of an insight? Okay, so Anonymous is saying which positions may hold similarities with other known sequences. So where are regions that are similar, right? So that is one of the questions. Marie is saying the similarity between reference genome and the other genomes or mutations. Okay, so how can we now quickly find those regions of similarity or dissimilarity between these genomic sequences? Great. Now, if I just asked you the question, how do I find mutations here? It would probably take a while, right? I would have to do some more analysis of this data, which is already the result of the analysis, but I have to do many downstream steps to actually get an idea of where the more or the less variable sections of this genome. What are the sequences that are grouped together by similarity or by difference? And how do I interpret the mutations that do have an impact on a particular region of a protein that I'm interested in and which ones are not, right? So initially, this analysis is hypothesis generating. I am starting to get ideas about what I could do with this information. And now I have to do some downstream steps of analysis to start getting answers to those questions. So now let me show you how this particular data set could be used to understand similarity between sequences, right? Instead of looking at particular regions per nucleotide or per amino acid, I can actually look at this phylogenetic tree. And in this phylogenetic tree, I can start gaining some insights about what am I looking for in the specifics of those sequence similarities. So here I have a number of groups. Group 1 group 2, group 3, and group 4, right? And so I can see that these groups, for example, group 4 seems to be the most distant from all of the other groups. Groups 1 and 2, though, seem to be fairly close together, right? And so based on this understanding, I can formulate the next question. Well, what makes these groups close together, right? Why are they closer together? And if they are close, but not the same group, what are still the differences between them? So I've refined my question from a general question to a more specific one, and I'm starting to ask the question, where specifically are they different? Okay. Yes, this works for any kinds of genomes. The time to analyze the genomes would take a while, but you would still get the results. So the same file that we looked at in Excel could actually be visualized in a more informative way. Let's take a look at this specific example using our sequence navigator. So please go with me to this link, and you will see the exact same data set that we're dealing with, but in a visual format. Okay, so when you get to this link, you want to go to data and select right here, comparison birth death coronavirus. Okay, so please let me know once you're on this page and you see what I see in the sequence navigator. Maybe put a number or yes in the chat box so I know that you're following along. All right, thank you, Swati. All right, excellent. So again, we see here the exact same information that we see in Excel, but instead of just looking at thousands of rows, we see this information in a very condensed way. And so based on what you see in this visual representation of that data, could you tell me where is group 1 and group 2? So again, I will open up this phylogenetic tree, okay? And let's take a look at the names of sequences. Okay, so here you see names of sequences are right here. And here you see similar names of sequences. So from looking at this phylogenetic tree, where I see group 1 and group 2 are closer together, what are those groups? You can hover over different elements on the screen and let me know, what are these two groups? Okay, does anyone have any idea? If I go over to the screen, right, and again, I can hover over here, for example, they're already organized into groups. What do I see? Okay, so somebody said protein S, so right here we have different, okay, so two groups, right? We have group 1 and 2. We know that they're closer together. What are those two groups? Leanna is saying SARS, okay. So here, indeed, I have the reference is SARS. But what are the two groups that you see closest together? Again, let me show you the phylogenetic tree. AY613947. So you can see that all of these AYs, 613947, what is this? This is SARS, right? And then what is group number 2? So this is, sorry, this was group number 2. Group number 1, let's take this one, LC522975. LCC 229975 right here, right? All right, great. So thank you, Vadivel and Leanna, right? So we have here SARS and COVID. So SARS-1 and SARS-2, COVID-19 and SARS-1 seem to be the closest ones. And so we have here MERS, we have here beta coronaviruses, And then we have COVID-19 and SARS-1. Now, beta coronaviruses are coronaviruses that cause the common cold. MERS was the Middle Eastern respiratory respiratory SARS, and that was a specific outbreak related to the Middle East and in, I believe, the Far East. So here we have two groups that seem to be closer together. Now, if we look at these two groups visually, instead of in the full sequence of what we were looking at before, where are the specific differences between those two groups, right? So again, these are the two groups, the pink and the red. They're the closest to each other. How do we know in which regions we observe the most variation? Okay, so now we're looking, instead of this bar right here, we are looking at this bar right here, right? And so here you have the reference, but in terms of the protein sequences. So these include the ORF1ab polyprotein, the surface glycoprotein or the S protein, ORF3a, envelope protein, membrane, ORF6, ORF7a, ORF8, nucleocapsid, and ORF10. So in which protein do we observe the most variation between group 1 and group 2? Does anyone have any idea about what could be that region? Now, let me explain some of these colorings. As you can see, each one of these represents a bin. These bin size are 500 nucleotide long. And the more variation is observed from the reference, the more red you see the color of this bin. So for example, if you look at this bin, the number of variations from the reference is 436 out of 500, right? So that's almost 100%. If you look at the gray one, the variation is 0. So there are 0 mutations in this specific bin. So if you look through these colors, where are the colors with the most variation from the reference between these two groups? Okay, so just type in the name of the protein that has the most variation. Any ideas? Okay, so for the ORF1ab. So here we have ORF1ab. Okay, IO is saying around 23,000. So around 23,000, that could be right here. Now, what is this protein? Okay, how do you get the protein sequence? So here you have the proteins, right? This is the reference genome. It's got the different proteins. You can click on it and see what is this protein. So for this, for example, is the spike glycoprotein. Here you have the ORF1ab. So just one, okay. All right, great, thank you. All right, we're getting some answers. So ORF1ab or surface glycoprotein. So let's take a look. So indeed, in ORF1ab, we're observing these particular changes, right? 213, fairly consistently 180, 207, and then here in the surface, we have 252. So if we are thinking about the total number of mutations in a particular region, because this region is so large, we do expect to find a lot more changes. However, if we're looking at a particular position that has a lot more variation in a very focused region, we are talking about the spike glycoprotein. Now, to understand, again, contextualizing our analysis with knowledge about how this works, we know that the spike glycoprotein is responsible for cell entry and therefore infection of the cell. And so if we think about the relationship between these different pathogenic viruses and their ability to cause disease, maybe one of the interesting areas for us to look at would be the spike glycoprotein. It's also important because in the context of prevention of the entry of that glycoprotein into the cell, we do need to look at the variation in the spike glycoprotein. So to understand the impact of this variation specifically in this position, we actually don't know anything else based on sequence alone. We now need to take from sequence into the structure, and we need to understand how does this specific variation can affect the structural properties of this protein. So let's go on and let's look at how this actually could help us understand those differences. Now, I clicked on protein compare, and this is our ability to now ask a downstream question. When we think of a specific area, a region on the sequence that has an impact on the structure, how can we find within that structure a particular region that has an impact on the interaction between two proteins? So here I can load the whole structure, or I can actually choose the different proteins, and I can also choose the different ways I represent this protein. But I've loaded here the whole structure of the surface glycoprotein, and I'm looking at the surface render of that structure. Now, this is a comparison between different hosts, bats and humans. And I can see here a specific non-random pattern that emerges, right? And so in this example, I can see that this pattern is actually represented by high variability in red, low variability or higher consensus, higher conservation with gray, and higher variability with red. So if I look at this sequence, I can see that there is a binding region in this particular red color that is affected, and also some specific positions inside this protein. Now, this allows me, as you have seen, to quickly go from sequence, relationship between sequences, to the impact on the structure, and ultimately to start thinking about the impact of the variants that I observe on the genomic sequence to the protein-protein interaction. Now, the question is, can I apply this kind of a visual exploration tool to a number of other problems that I might be thinking about? Let me give you an example. So we went from the platform where we have access to a number of tools that, instead of looking at various types of scripts and solutions that solve a focus problem, combine different scripts into a rapid workflow that you can execute to prepare data for interactive exploration. This is important because we have such an abundance of genomic data that is highly variable, and that allows us to ask questions about practically anything, right? And inside those available sequences of genomic data, we need to understand, here's an example of variants of concern. Here we have an example of the types of sequences that we can find, right? So this is NCBI virus, where we can literally find information about any type of virus that has been sequenced. We have a lot of variability with collected data that we already discussed that can cause bias. And nevertheless, we tend to think about that variability to kind of detect these major trends, right? So what is being reported today, a lot of it in the media, is how the general variability between sequences or particular variants of concern emerge in different places around the world. Now, if I think about how I would want to textualize this type of data analysis with the available data that I have, I would want to focus it and develop a custom solution that allows me to ask much more focused questions based on the available data and based on performed analysis. And so that brings me to the next important question. Genomic data represents huge data sets with variable properties. And those variable properties are based on the type of data that was collected and by whom, when and where and how it is stored, and how it was annotated with important metadata that allows me to ask specific questions with a level of reproducibility and reliability. And so that is why when we put together these analysis pipelines, we're essentially trying to make them logical. And we're trying to make sure that the data that we put into the pipelines come with suggestions for analysis. So here you can see, as we've seen before, this guided process. This guided process allows us to put in the data and allow the platform suggest some kind of an analysis that essentially leads to color coding that is logical, explanation of methods so that as I analyze the data, I can learn about the specific methods that I'm using, providing different types of inputs and outputs that I can visually explore and ask downstream questions, and essentially leads to a guided and reproducible workflow that I could then apply to specific data sets that I have designed around a specific question. And so this is consistent across these many different areas that we have already seen. Now, when we go from doing such analysis to an actual insight that is actionable, we want to design a context. So here, historically, you can see the context of different types of interventions, such as vaccines, antibacterial drugs, so antibiotics. You can see different preventive measures that have been effective in reducing the prevalence. And you can see different types of pathogens that have caused disease over time. Now, given the variability of approaches that have developed over time, we live at a time when genomic data availability is making it possible to start asking very specific questions. But instead of looking at general trends like this, we need to design very specific questions around specific geographic regions, about specific pathogens, about specific data sets that we have access to start formulating very specific data sets with analysis methods that will provide us with actionable results. And so when we look at this tool that essentially was designed to contextualize data, we are looking at an example of streamlining this approach, right? Making it faster, translating the raw data to actionable insights in a much faster way. And so what we are proposing to do with this tool, and we're inviting all of you to participate in this effort, is to start designing specific questions around specific pathogens, geographic regions, and challenges like vaccine design, drug design, or understanding of the basic mechanism of disease that could be answered with this tool. So I'll just give you one more example, and then I will pass it on to Mohit to talk about how you can also start generating project ideas and research questions that could lead to some of these important insights, actionable insights that could lead to the improvement. This is another variation of the same tool. You can see the same concept of bins. Instead of samples, these are passages that allow us to study the impact of frequency of mutations on variability in the viral quasi-species that tend to be associated with virulence. So the same is known about many different pathogens. If you think about the immune system, that actually makes a lot of sense. If you have one type of an enemy that consistently attacks in the same way, you can easily identify and neutralize that enemy. But if your enemies are varied and they attack in very different ways, and you have to adapt to each one to address the challenges that they pose, that spreads out the immune system thin, and therefore the infection overall in one place will be able to breach the defenses. And so here you can see an example of poliovirus rate of mutation variability based on specific positions in the reference genome and the associated proteins that are being produced from these polyprotein sequences that allow us to quickly navigate this complex data set and start asking the question about what types of mutations we are seeing and how we can see the specific variations. I'll just turn this on right here, that could be detrimental or beneficial to viral replication, right? So for example, here I can see a region with detrimental variation. So I can start asking the question, how does this specifically affect viral structure, right? And so a lot of the connections that I'm trying to make are going to be very obvious if I use this multi-layered integration tool to essentially combine the different types of data that I have into a single exploration environment where I can rapidly go from raw sequencing data to very specific types of insights that I could then leverage to understand the mechanism of disease and to translate my understanding of this disease happening to an intervention or a prevention strategy. So as I mentioned, these types of questions are being asked by participants of our OmicsLogic training programs. And one of these programs is focused on malaria and COVID-19. So in this program, we are going through different types of genomic data, different methods of analysis, and ultimately designing questions that will help ask the specific data sets, specific types of questions that are important to understand those pathogens. So to talk about that more, I'm going to invite Dr. Mohit Mazumdar to take over and give you some examples of student projects that they are working on. But before he takes over, I want to ask if you have any questions so I can answer those right now. Okay, so I'll be here and you can continue posting your questions in the chat box. I will paste it on, pass it on to Mohit to talk about those projects. Thank you.
Thank you, Ilya. Let me share my screen. Okay, hello everyone. I'm Mohit Mazumdar, and as a co-mentor and project guide, every day I have been able to work on exciting and this dynamic field of bioinformatics and data science. And so far been able to publish 30 bioinformatics projects in some of the peer-reviewed journals. And currently our team has been working on more important bioinformatics projects in collaboration with several research labs that are practicing translational sciences. So to talk more about the research fellowship, I just wanted to show you a slide here. So about the research fellowship or the independent project, the journey to completing an independent research project is really complex, right? You first learn about a particular topic, and then you review data sets or databases for specific project, data sets, and then acquire the skills to analyze that data set, right? So even in the established educational system where this is going on as a part of their PhD program or a master's program all around the world, this process can take like years, and it is like dependent on several factors. And some of those factors are like having the right faculty, resources, and the mentors that can guide this process. So there are questions, I guess. So what I wanted to tell you is that this OmicsLogic Research Fellowship is really a cool opportunity for many students and graduates to be able to develop and complete an independent project. So this research fellowship program is a structured program that allows participants to work on self-paced and a flexible schedule and work with a team of experts on cutting-edge topics in healthcare research. So some of the examples here, as you can see, the participants started learning from the workflows that allows them to learn about complex bioinformatics pipelines and algorithms and their outputs. So this,
Mohit, I don't think your slides are showing the way you intend them to.
Okay, yeah, it was, I think I have to then log out and come back. But I just wanted to tell you, tell everyone about the research fellowship program. And one of the major advantage of this program is to, you'll be able to communicate the outcome of this research and get feedback from experts uh, from experts and peers to improve the project by including information from literature and the analytical steps. So most of our past participants have gone through this entire process of starting from the training and next to work on example datasets and then exploring the publicly available datasets to come up with research questions and then work on the research questions and then complete it into a poster conference presentation. So here you can see a poster on RNA sequencing and use of RNA sequencing in lung cancer. So similarly, we have been working on several research areas, as Ilya rightly pointed out. So we are working on infectious diseases, we are working on cancer genomics and agriculture. So it depends on the research question being asked by you. So we as a team are trying to help guide this entire process, because this is like an opportunity that you can work on as a part of your thesis, as a part of your own interest to develop something of of your own interest. So these materials and these entire process is being reviewed, and it's quite it has been quite effective and transformational. And to talk more about like the upcoming opportunities and what we will be doing next, I would like to invite Ms. Deepsha. So over to you, Deepsha. Thank you,
so
everyone.
much.
And if you have any questions regarding the research fellowship program, please feel free to reach out to us. So I'll be putting my email address. And if you have already a research question in your mind that you wanted to discuss to learn about the possibilities of doing that, whether we can be able to work with the data which is publicly available, please feel free to reach out to me as well. So I'll be putting my email address in the chat. And thank you, everyone.
Thank you so much, Dr. Mohit. So I will go ahead and walk everybody through this entire process. I know that everybody has a question. How can I join a program? How can I get started? So let us look at everything one by one so that we are able to go ahead and start with it. So the first platform that I welcome everybody to get started with, this platform, the registration to this platform is free. So what you do to register on the learn.omicslogic platform, it's a learning tool or an educational portal. So you go to login right here. If you already have an account, you can sign up or sign in with the same email ID. I already have an email ID and account with my email, or you can also log in via any of these social links. So please feel free to create an account along with me right now so that I can introduce you to some of these courses and resources. So if you come in here, there you would come across some of these courses, and we have designed them in a stepwise manner, but not necessarily that everybody has to start right with the first course. And these courses have lessons which are a mix of open or free lessons, while some of them are lessons that you have to subscribe to or which are available to as part of all of our OmicsLogic programs. So is everybody there on learn.omicslogic? Can you put number 1 in the chat box so that I know that you're here? Okay, Io and Lena, thank you. Anybody else here? At least 5 people who would be able to sign up. Thank you, Swati, Gulam, Anonymous. Right, so we have 5 people who have already registered. And please note that these courses which are not marked with a star are completely open, completely free for anybody who is coming in and wants to get started today and right away. So great that everybody is starting with the courses. Similarly, you will have a course on Introduction to Bioinformatics. Then there are several other courses. Now, today's session, we discussed a little bit about genomics. So if you come down along to the genomics lesson, you would start learning from DNA structure and variants, then working towards getting introduced to how to work with DNA sequences in R, then getting an introductory overview of what genomics is all about, and then you progress to these lessons. So here you would find that we have provided you courses which you can access like this by signing up for free and get started right away, right today. So please feel free to explore anything, any of the topic that you have an interest in. And if you have any feedback for us, please reach out to us at marketing@pine.bio and share those feedback with us. Yes, so Ramavat, if you have interest in genomics, please go ahead and take the genomics course. We welcome everybody to go ahead and check these courses. And we also have a few projects here as well, and we keep on adding more projects here. So these projects are taken from peer-reviewed publications, and what they help you exactly is by learning this entire project process. So you're able to see from these projects, like for example, if I'm to look into the Ebola virus project, you can see like what is the introduction to this entire Ebola virus story right here? What are the bioinformatics methods utilized? And in conclusion, and you would have to do some kind of hands-on to learn more about how this project has been executed. That gives you a better idea on if you're planning to work on a project of your own. So these are some example data sets or example projects that are available. And of course, the data sets are also provided along with them. And we also have a full set of courses on R and Python. Now, the R and Python courses have code blocks within them, which means that you do not need to install R or Python in your computer. Ramavat has a question. How should I register for the course, ma'am? So as I showed you, you have to log in to learn.OmicsLogic. And once you log in, you will have access to the courses. Now, I will talk about that. You're seeing that there are a few lessons which have a star sign marked as a premium lesson. So how do you get access to that? Those lessons or the full set of courses available on this platform comes as part of our Omixlogic programs, some of which we talked about today, like the Bi-Informatics for infectious diseases, the genomic program, which is ongoing right now, and the upcoming Transcriptomics program. So I wanted to actually take you through the upcoming programs that we have and talk a little more about each of these programs right here. So let us take a look at these upcoming programs. So we keep on updating this page. So anytime you want to join or check which programs are there, you can go ahead and check this. So the first program is a Bioinformatics Research Fellowship. This is the research project program. And this program is available all around the year. And what we usually do in this program is a research fellow joins for either a 3 months or a 6 months period. And these are the typical timelines that we have, and we provide for you to go ahead and develop your own project, as Dr. Mohit was speaking about earlier. So in the 3 months program, is recommended to anyone who has an idea of developing a project but is not sure how to get started. So for the 6 months program, we recommend it to anybody else who wants to work on a longer term in the project, in the means of that they want to take the project forward to a poster or a publication, though that depends, of course, on the complexity of your project, on the time that you're available to give for that project. But we typically have seen that our participants are able to come up with a project, even high school students, in very short period of time, which is like 3 months. And for this program, we typically recommend at least 6 to 8 hours per week to be able to come up with that project. So the program starts with training, and then we work with you by assigning you a mentor from our team who works along with you in helping you develop that project. So you go through an intense training session And this project, please note, that is your own project. We do not offer you the topics, or we do not provide the topics. You are free to choose your own topic. What we do is provide you that mentorship where you're able to understand whether the project that you have chosen is going to have some reproducible results, whether that project you can apply it in different places, like for publication, or if you want to submit a paper for review, poster presentation. So we work with you through this. And here are some of these examples right here of projects. So I'm also going to be sharing this link in the chat box for everybody. And you would see that these projects are done by a mixed group of people. By mixed, I mean that participants have come from high school, from research level, from undergraduate, masters, PhD level. So this is really like for everybody. Whoever is interested in doing a project, you can actually go ahead and watch these video sessions, which are recorded by the participants themselves, where they speak about their own project so that you can learn about this entire process. So I would leave you all to explore this. Now, the next program that we offer, and which is coming up right now after this, is this program in collaboration with All India Institute of Medical Sciences, New Delhi. So we have this workshop on lymphoma, and you can register for this program by filling out the form at the bottom of the page, which I'm going to be coming to next. Then the other program, which we discussed briefly in today's session, is the Omixlogic Transcriptomics program. So this is also part of the Bi-Informatics for Infectious Disease program that we have been discussing today. So this program is starting now on April 16th, and the registrations are open to join this program right at the moment. So you can go ahead and check this program as well, as you understand that this program will have transcriptomics for infectious disease research. And we also have another program that is starting in India with a lot of our collaborators, and this is on biomedical data science and drug discovery. So you can also join this program. And so how do you join either of these programs? You have to fill only one form. And once you come down here, you can choose which program you are interested in. So you do not have to fill multiple forms. You can choose right here. And even if you want to apply for multiple programs, consider filling out the form twice so that you're able to submit it twice, and you will receive details regarding this. In case any of the details are not very clear to you again, or you want to ask us more questions, please feel free to again reach out to me, marketing@pine.bio, and I would be able to clear those out for you. So Abu Kari has a question here. I would like to address it at this moment. Are the Python coding courses paid? So the Python coding courses, again, some lessons are free while some lessons require a subscription. So if you can get the recording of the session, so I think Daniel, yes, we will be sharing the recording of the session, of course. So yeah, so I think that I have answered the question. Now on regarding access to the Tbio info platform. So the programs that we have mentioned here all come with educational access to Tbio info, which means that whatever cost that you're paying for each of these programs covers the cost of the Tbio info license. But in case you are interested in just exploring the Tbio info platform, we also have another licensing here, which makes it clear for you how to get started with getting access to the Tbio info platform. It's called the Gene Lab. For researchers, we offer a research license where you're able to work with your own data at $99 per month. But for first-time users, you have to take this license for 3 months, which is $297. So that is the research license, while the educational license, which comes with all of our programs, costs $75 per month. while all of the programs already include this in their pricing. So anybody who joins any of the OmicsLogic training programs will have access to Tbio info. The access would be created by us and sent to you. So if you have any questions regarding that as well, we would be able to help you with this. So let us know if you have any questions about this, and I will be able to help you. And while you are here, I wanted to welcome you to join us here on LinkedIn and some of our other social media networks. So we have this group in LinkedIn where we keep on sharing many interesting posts, and not just posts, we also keep sharing job opportunities as well as any other things which are relevant to the bioinformatics community that we have. So please feel free to go ahead and join this group. We also have a group in Facebook. So please feel free to follow us on Facebook as well. And here is our Instagram page. So we also keep on sharing all of these updates on Instagram, as well as do polls sometimes on our stories. So please go ahead and follow us there if you are more active on Instagram. And now again, I would like to go ahead and ask if you have any questions so that I may answer them and we can then take those questions. So thank you all again. And while we are going towards the end, what I wanted to mention here is that we also offer scholarships for our programs that any participant can avail. These scholarships are given to participants with whom we have an interaction. So the process of the scholarship is this, that you fill out the form at the bottom of this page. You need to mention your references from your college or university who go ahead and join that meeting with us. So any one of your references can get on that meeting with us. And once we have spoken to the reference and understood why you would need that scholarship, you would be receiving all the details from our end. So you can utilize this option for any of our programs that are here. So please feel free to post them down and fill out that form if you think that you require that scholarship to join any of the programs. And I hope that this was clear to everybody. So again, if you have any questions, if you want to reach out to us, we have the email ID marketing@pine.bio. We have also shared Dr. Mohit's email ID, so you can also reach out to him for any kind of research question that you may have. So thank you all again for joining us today. Yeah, thank you so much, Mari. Okay, it's wonderful to have you all here. We'll share the recording and all of the links to today's session via email with you all. And have a lovely week ahead, everybody. Thank you again.